Hi, everyone. Sean Egan here, Chief Scientific Officer from the family's SCN2A. We're really excited to have Michael Coulter with us today. Michael Coulter is at our most recent Families SCN2A conference. He won our Best Poster Award, our inaugural Visionary Award for Best Poster for the work that he presented during our conference. Michael Coulter is an MD PhD from UCSF, and we're going to have him today go through his poster. Michael?
Great, thank you, Sean. Yes, so I'm excited to talk a little bit today about the work that we're doing on a rat model of SCN2A loss of function disorder. And I will just share my screen to go over the details of the project that we've been working on. Okay, so the goal of this project has been to model SCN2A loss of function disorder in rats. And there are many different rodent models of SCN2A loss of function disorder, typically in mice. And the reason that we're excited about modeling this disorder in rats is that rats have two features that are different from mice. First, they learn spatial memory tasks more quickly, and they can learn flexible tasks more easily than mice. And we think that this is a good model for learning in humans. And second, rats are larger in size than mice, and this allows us to record the electrical activity from the brain more easily and from more cells in the brain. So I will just jump in. And the goal here is that we are trying to model learning impairments, which is one feature of SCN2A loss of function disorder. And to do that, let me show you. Okay, so if you start here, where it says spatial alternation tasks, what we're having the rat learn is to alternate between three locations that give reward. In this example, the arms labeled 3, 4, and 5 give the animal reward. And the animal has to learn a alternating sequence to get the maximum reward. So when they're at the center, they have to go to the left arm. Then when they're at their left, they have to return to the center. And now when they leave the center, this is sort of the most difficult decision. They have to decide whether to go to the left or the right. And that is determined based on where they were before. So if the rat previously came from the left arm, now the rat has to go to the right arm. And then from the right arm, the rat returns to the center. This task is exciting to us because rats can learn it relatively quickly. And this task has been previously associated by work in our group with the function of the hippocampus, which is an area in the brain that's important for learning and memory. And how the rats perform this task can tell us a little bit about what's happening in the brain. In particular, there are two different types of errors that animals can make in this task. One is an inbound error, and that is on trials when the animal is returning to the center. The other type we call is an outbound error. That's where the animal has to decide to go to the left or the right. And what we've found previously is that the hippocampus is particularly active when the animal is deciding whether to go left or right. And what's really exciting is that when we looked at the animal's behavior on this task, we found that SCN2A loss of function rats had a specific impairment in the outbound trials, and that's shown here. So the top of this plot here is the inbound trials, the errors on those trials, and you can see that the errors decrease over time. And here black is wild type and red is SCN2A loss of function, heterozygous. And there's no difference in the inbound errors between the SCN2A loss of function and the wild type. However, in contrast, you can see that the SCN2A loss of function rats make more outbound errors. So that means that they're having a harder time deciding whether to turn left or right compared to the wild type animals. And this is exciting because this suggests to us that there's perhaps changes in the hippocampus that might underlie this behavioral difference. Interestingly, we also found that in this task, when the animals are switching between different versions, the SCN2A animals switch more slowly. So if you look back here at the main 6-arm environment, we can sometimes have the rat do arms 3, 4, and 5. And then we can switch it so that the active arms are, for example, 2, 3, and 4. When we make this switch, the SCN2A loss of function rats switch more slowly than the wild types. And that's also an exciting finding, suggesting perhaps a impairment in task switching. So then the goal of this is to learn about what might be happening in the brain that could underlie this change in behavior. And based on the increase in the outbound errors on this task, we think that there's perhaps a defect in what we call awake hippocampal replay. And this is a process in the hippocampus, a part of the brain that's important for learning and memory, that enforces previously learned experiences. So basically helps the animal associate previous reward experiences with learning what to do better in the future. And so we're now looking at, just a second. There we go. We're now looking at awake hippocampal replay in the SCN2A loss of function rats. And what we can do is we do a surgery where we implant electrical recording wires into the brain of the rat, and then we can assess the activity in the hippocampus. And what we found preliminarily is that there is less of a particular signature in the hippocampus called awake replay in the one of the SCN2A loss of function rats compared to wild type. So this is preliminary data that we've only looked at one pair of rats so far. But what you can see in this plot is that the frequency of these replay events is lower in SCN2A. Those are the blue bars, the SCN2A loss of function, compared to the gray bars, which are the wild type rats. And we're now looking at this in a larger cohort to see if this difference is consistent. What's exciting is that this difference would actually explain the difference in learning that we saw during the task. Another advantage of our system is that we're also able to look at what the brain is representing, sort of what the rat is imagining during these recollection events. And so this plot here shows an example where if we look at one of these sharp wave ripple events, we can look at which cells are active in the hippocampus and see what they represent. And in this example, there's the rat is sitting at the end of one arm, and this red arrow shows that the representation in the rat's brain is actually in a different part of the environment. And this is exciting because this helps us understand what the hippocampus is doing during learning. And if we can find differences here, this might help us understand what's different in the hippocampus of the SCN2A loss of function rats. We've also been looking at this at the cellular physiology level in the hippocampus. This has been in collaboration with another SCN2A investigator, Kevin Bender. And there we also find that there's a impairment in synaptic physiology, synaptic potentiation in the SCN2A loss of function rat. And that could also contribute to the behavioral differences that we're seeing. So in summary, this work is exciting to us because we found a consistent learning impairment in the SCN2A loss of function rats. And we think that this can be used to help us test the efficacy of future therapies, for example, gene therapy through a process called CRISPR activation. And our prediction is that maybe gene therapy would improve this learning impairment, and then that could be applied in a human trial where similar sequence learning changes could be measured as an outcome of the trial. So thank you, and I'll stop there. You're muted.
Always happens, always happens. But thank you so much for all the work. Really interesting science. Maybe can we put the poster back up one more time?
Oh, yes.
Maybe we can talk through a few of the elements. And I want to be cognizant that we will have families kind of reviewing this too. So maybe to kind of recap from 10,000 feet, in loss of function, SCN2A variants with loss of function, there's some learning deficits, and you're able to test that in an animal model, rats, and then kind of get into the weeds on actually figuring out why that might be going on.
Right.
So you mentioned these spatial tasks. we can't give rats like a math exam to test like their IQ and how they're learning. So these spatial tests are kind of a way to kind of give the rats a test.
Yes, exactly. So these spatial alternation learning tasks, the rat is required to learn a sequence of events in order to do well on the task. And so we think that that could be similar to sequence learning in human development, and that is similar to, for example, learning how to do math or how to read.
Perfect. In the next section, you mentioned inbound and outbound. Why do you think there was a difference between the inbound and outbound rates? Why was there less of a difference in outbound? Is that just kind of a nuance of the experiment, or any ideas there?
Right. So we found that there was more impairment in the outbound trials in the SCN2A loss of function. And those particular trials require activity in the hippocampus. In particular, these sharp wave ripples that replay previous experience and are important for consolidating previous experiences. And so we think that this impairment on the outbound version of the task suggests a specific a specific memory impairment when SCN2A is absent. So this suggests that the animals are engaged in the task and that they may have a specific impairment in memory for guiding their learning. And that may be an indication of something specifically that's missing when SCN2A is absent.
Gotcha. I noticed on some of the graphs that the loss of function het mice are able to learn, but it just takes a little bit longer. And that is something we do see in our loss of function kiddos, and that they are able to kind of continue to learn, but the rate is a lot slower. So is the hope that, if we're able to have a corrective therapeutic, that the rate of learning would then be able to increase?
Yes, exactly. The prediction is that if we could restore the SCN2A expression level to normal, for example, through a gene therapy like CRISPR activation, that the rapid learning would be restored and the learning would be faster.
Terrific. One question we always get whenever a project is loss of function or gain of function centric is, would it work on the other side? Like, is there any potential for this type of work or learning experiments to be done in the gain of function mice?
Yeah, I think that's a great question. And I think that is something that we could look to do in the future. I think with advances in CRISPR techniques, we would be able to generate, for example, a rat model that would model one of the more common gain of function variants from humans, and then we would be able to do similar tests in that model.
Fantastic. When we're always so focused on getting, new treatments and cures, and a big part of that is, getting medicines into clinical trials. And a clinical trial is almost like a big test, and an important part of that test is like what's the endpoint, or what are they testing? Hearing your work, it seems like this has, could have a lot of implications for that, as they're evaluating new drugs and new molecules. What should they be looking at? I think sometimes they look at seizures, but your work suggests that they could be looking at learning as a potential endpoint. Do you have any thoughts on that, how it could be done, or whether that's an avenue that should be pursued by drug developers?
Yes, I think that's a great question. And yes, I think our hope is that this kind of model would hopefully be able to show that an intervention could improve learning, for example, spatial learning, which is one of the things that we measure, for example, in some of the developmental tests like the Vineland. And the hope is that if we could show an improvement in learning with therapy in the animal model, like the rat, then that could be used as an outcome in a human clinical trial.
Terrific. I noticed that you use a rat for most of your experiments, and sometimes researchers will also use mice. Could you maybe talk about why rat is a good model for these experiments?
Yes. Yeah, that's something that we're really excited about, is using rats. And for us, there are two main reasons. The first is that we have been able to find a consistent learning impairment in the SCN2A loss of function rat. And that's something that has been more challenging in mice, where some mouse strains have learning differences and some don't. So we're excited because this is a more consistent learning deficit, which will make it easier to use in modeling in a preclinical work for a clinical trial. The other reason why rats are exciting is that they're much larger than mice. And so that allows us to do more detailed recordings of the activity in the brain that can help us better understand exactly what is different in different parts of the brain.
Perfect. Well, I think that's any questions for us, Michael?
No, I think this has been a wonderful experience, and I'm really excited to work with the foundation. And it was wonderful at the meeting last week to meet so many of the families to really get a nice personal touch for where this research is headed. And that really helps motivate researchers like me to do this work. To meet the patients is really amazing.
Phenomenal. Well, congratulations again, Michael, our 2023 Visionary Award recipient. Thank you, Michael.
Thank you. Thank you.